Hello biologists, we're going to talk about the end of Unit 2 test for MNVA Biology. It has 13 multiple choice questions, and for our test there are 4 essay questions. These are tough. Your peers wrote one of them for each section. So you are lucky enough to get that extra question written by your peers. Just a reminder, it's spring here in Minnesota, and we have an example of one of the conditions that you need to cause evolution to occur. Hardy Weinberg, lack of Hardy Weinberg stability happens when you get sexual recombination. And right now, or in a few weeks maybe here, as our spring is a little bit late, maple trees are going to start flowering, and flowering is an example of sexual reproduction in trees even, and you didn't think trees reproduced sexually, but they do. So pay attention to around you as trees start to flower, and that is an excellent example of variation that happens through recombination in sexual reproduction. So let's talk about the Hardy Weinberg model and the 5 conditions for Hardy Weinberg stability. No migration, no mutation, random mating, no natural selection, and a large population. Think about the fish lab we did in 2.13 with goldfish. The easy way to remember these conditions for Hardy Weinberg stability are to remember M&Ms. You've got 3 M terms here: migration, mutation, and random mating. So migration, mutation, mating, and if you think about a dish of M&Ms here, this will help you remember. If you decide to eat all of the blue M&Ms, that's a form of selection. Hardy Weinberg stability is no natural selection. So don't do this if you want your M&M population, the allele frequency of your M&M population, to remain stable. And if you have a large population, you're less likely to have natural selection happen or allele frequency to change greatly than if you have a very small population. If you decide to eat all the blue M&Ms in this very small population, you're drastically going to affect the allele frequency. This is the only blue M&M in this population. So remember your M&Ms and see if you can fill this in. No mutation, no migration, random mating, that's if you want stability, and no evolution, no natural selection, and a large population. Because if you have a small population, it's going to be very easy for allele frequencies to change very quickly. Now you can start eating yellow M&Ms and change that allele frequency. In the lab that we did with goldfish, we didn't have Hardy Weinberg stability. We changed the allele frequencies across the lab. Remember, you were a hungry shark and you were eating only the pretzel goldfish, or excuse me, only the gold cheese goldfish, not the pretzel ones. So if you as a predator are selecting for a particular allele, you're only eating the goldfish because they're easy to find, you're not eating the pretzel fish, then that's going to cause natural selection. Let's look at this graph for a second and think about variation. The variation of the mass of watermelons. What's going on here? Is there a lot of variation? Is there a little bit of variation? If we only saw a little bit of variation, this curve would be even narrower. It'd probably just be over the 6 or 7. If we saw a lot of variation, the curve would probably be more shaped like a dome. It's kind of more of a hill shape here. So we're seeing moderate variation. A good answer for this would be B. Now let's go back to our predator question. A predator eats one phenotype rather than another for a whole year. What will happen to the allele frequencies? Well, here's the data from our experiment with goldfish versus pretzelfish, and we saw the gold allele go from 0.77 all the way down to 0.44 at the end of the experiment. So we saw a change in this allele frequency. So if a predator is selecting for one phenotype, for one particular allele, there is going to be a change in the allele frequency. We are not going to see Hardy Weinberg stability. We are going to see evolution. There are other factors that can cause natural selection besides predators. We had a shark in one experiment. We have wolves in the second natural selection experiment, Lab 2.20. Other factors like sexual selection can make a difference. These two walruses are battling it out to see who will get to mate with the females in their group. Ladies in certain bird populations, the female birds, choose male birds with dramatic characteristics. It can be big neck pouches if you're talking about prairie chickens, or long tail feathers in many species. So the choosiness, or the battling it out between the males, can affect the allele frequencies. So mating is another thing that can cause natural selection. In both these cases, this is non-random mating, and that's going to affect allele frequencies. If you have random mating, that's not going to affect. If ladies are just like, I don't really care how long your tail feathers are, then that's random mating. Non-random mating, which would be picky females or males battling it out. That non-random mating causes allele frequencies to change and evolution to occur. So who chooses their mate or who gets to mate in the long run can affect allele frequencies. Lethal alleles are alleles that will kill you if you get 2 copies. But they're maintained in populations over time because remember, you need 2 copies. You can still get a copy without the mutation being lethal if you from a heterozygous carrier. You get only 1 copy of the lethal allele. So in this case, if double F in fish, let's say that's a gene that causes your gills to malfunction completely, if you get 2 copies, you're a dead one. If you get a heterozygous situation, you're fine, and of course you're fine here if you get the dominant OK gill allele. So this is how you would pass on that lethal allele. This fish wouldn't survive. So here's the heterozygous condition, and heterozygous carriers. Answer C is correct here. How is a trait maintained in a specific population? Well, in order for the allele to get passed down, there has to be mating going on. And so your parents mated and you inherited their traits. So you can inherit it from your parents, but in order to mate, your parents had to survive to reproduce. They had to survive all the childhood diseases like measles, and mumps in order to get old enough to find a mate, marry, and reproduce. So here you go. E is the most correct answer. Taxonomy is about, and we talked about taxes. Taxes, you need to be very organized to do your taxes. So the word tax in Latin means to organize. So if we're going to do taxonomy rather than phylogeny, we're going to talk about classification. And to talk about classification, these 2 categories were added most recently. Domain was added because of archaea, the bacteria that belong in archaea, because they are very different. They're actually more related to Eukaryota than they are to the Monera domain. And life, because if we're going to classify viruses, they don't belong with other living things. Viruses are not considered to be alive. Taxonomists focus on similarities and differences with other organisms. That's how they organize organisms. Remember, tax means to organize in Latin. If we're going to talk about the higher order thinking questions here that are upside down on all the slides, DNA is important to phylogeny because phylogeny is about relationships. Remember, family and phylogeny sound similar, so phylogenists think about relationships or family structures. If an organism is in the domain Eukarya, what does it have in common with other organisms in that domain? Well, there's a huge hint in this picture here. The structure that you're looking for is dyed blue. Right here in the middle of these cells, there's a nucleus, and that's what makes you a eukaryote. The two domains that do not have a nucleus are Monera and Archaea. And Archaea is actually more closely related to Eukaryota. So we're going to talk about predators and their impact on natural selection again. If predators started eating lizards with the longer tails because they could catch them more easily, what would happen to the allele frequency of this tail gene? If this guy, because he has a long tail, doesn't survive to reproduce, what's going to happen to the frequency of that long tail gene? I'm guessing the long tail gene would decrease. And if the long tail gene increases, there is going to be proportionally more lizards with short tails. So the short tail frequency would increase, and both B and D are true. This situation would not represent Hardy Weinberg equilibrium because a change in allele frequency is happening. These are related to your essay questions. Could the allele frequency change happen in a population if predators use monkeys' tails to catch them? Could the same kind of change occur? I don't know. You'll have to think about that one. In this population, P and Q were changing. This is the Hardy Weinberg equilibrium allele frequency. What is true if P and Q are not changing? If these don't change, you have 0.77 or pretty close to it throughout the experiment when you're monitoring the allele frequency over the population over time. If that doesn't change, you have a stable population and you're in Hardy Weinberg equilibrium. If it doesn't change, it's stable and you're in equilibrium. Let's talk about the levels of classification. Circle the most inclusive levels of classification. Which levels contain the most organisms? Is it domain, kingdom, phylum, class, order, family, genus, or species? Which some people remember by Dancing King's play chess on funny green squares. Here's your Dancing King, and here are your funny green squares. And if you guessed domain, you are absolutely correct. Let's look at the two organisms that are closely related, or two of the organisms that are closely related on this phylogenetic tree. This represents, it's a kind of a big view of all life. This would be the single cell organism we think all life evolved from. And we're looking for two organisms that are really close together. If they're going to be very closely related, these two are a good example because they branched off very recently. This axis here, it's sort of a graph, is time. So this would be a long time ago, and this would be more recently. These two organisms are probably quite closely related because they branched off recently, even though you don't think of animals and fungi being closely related. They're more closely related than, say, bacteria and fungi. And now we construct phylogenetic trees using DNA evidence. some of that DNA clock stuff that was discussed earlier in the unit is really important in constructing these.